Welcome to the a-plus-hsc Biology series by Nelson. My name is Tim Sloane, one of the authors for the biology text. In the series, there are two products. On the right there, we have the study notes that I was involved in the writing of, but importantly on the left-hand side there, we have the complementary book, which is a whole series of practice exams and HSC style questions, which will really complement the study notes booklets well. So as we move through the presentation, I just want to take you through some of the top tips in preparing for your exams and ensuring exam success. In terms of preparing before the HSC exam and certainly before your trial HSC exam as well, some key tips are that it is absolutely essential that you know your NESA verbs, the glossary of terms, and I'll certainly take you through some of the key terms as they appear in the biology exam. Obviously, but surprisingly not necessarily done all that well in some cases, you must know your syllabus and you need to be able to break down the knowledge and understanding components of the course, but importantly, the working scientifically skills. And a second document that many students aren't as familiar with are the performance band descriptors, and they basically give you an indicator of where you are traveling at any point in time, but certainly give you a real key indicator of what you need to aim for if you wish to move up the bands from a band 3 to 4, 4 to 5, and 5 to 6. And again, one of those key tips is you analyze those performance band descriptors to get a sense of where you are sitting at the moment, and then look for those key descriptor terms that appear in the band 5 and 6, for example, and know what you need to do to aim for those higher bands. Goes without saying, it's essential that you study and make your own notes as they relate to those content descriptors for the knowledge and understanding and working scientifically component of the course, and not just copy down notes from the board or from the textbook. It really is essential that you really understand the content and know how to apply that to written responses in your exams. Which then leads me to point 5, practice, practice, practice previous HSC exams, and don't just do the multiple choice because they're convenient and easy just to lock in one of those 4 responses. You must really engage with those extended response questions, and importantly, mark your own responses using the allocated marking criteria. So I mentioned the key NESA verbs. Now, obviously, there are a lot more than the ones that appear on your screen, and I've intentionally highlighted the explain verb for the following reasons. It's a rough estimate, but around 60% of the HSC biology examination paper will be pitched at that explain level. And so you really need to understand what it means to explain key biological concepts, and basically make sure that you don't fall short of that, because where students typically lose relatively easy marks is they'll give those superficial responses at the identify and the describe level. And if you were to sit down and have a conversation with your classroom teacher, quite often they can extract that necessary level of an explain out of you verbally, but many students actually fail to, I guess, commit pen to paper and in terms of getting the necessary marks for an explain question. And then similarly, those really high order questions will be pitched at that analyze, assess, and evaluate level. And so again, it is absolutely essential that you know the definitions of each of these verbs and know the necessary level to pitch your written responses at. So I did mention in the introduction the performance band descriptors. I'm only going to talk through one of these, that being a band 6. Hopefully you'd all love to be aiming for that band 6 level. But there's some really important keywords contained within the band 6 descriptor that I just wanted to draw your attention to. So as you read through this with me, just have that internal dialogue with yourself and ask yourself, for example, can you clearly articulate the difference between accuracy, reliability, and validity? In the middle there, those middle dot points, you can actually see that it's mentioned 3 times actually, accuracy, reliability, and validity. And one of the key no-nos is not to use one of those words in defining the other. Okay, so at a band 6 level, students really know how to articulate the difference between those terms and be able to apply them to both first-hand investigations and the extraction of second-hand information, for example, from peer-reviewed published journals. Now, another key set of terms that really jump out at me in, I guess, the blue aqua highlight there is the terms trends, patterns, and relationships. So there are actually several syllabus dot points that explicitly mention those terms, trends, patterns, and relationships. And I, again, ask you at that band 6 level, can you clearly articulate the difference between those terms? And if not, you are certainly not currently at that band 6 level, because again, it's a key distinguishing feature of a band 6 student. If you were to go back and engage with the band descriptors and look at what constitutes a band 4 and band 5. Now, a couple of other really important points, just in that first point there. Students demonstrate extensive knowledge and understanding, and importantly, they can apply that knowledge to complex and abstract ideas. And then if I then jump down to the very bottom point, students are able to apply that knowledge and information to unfamiliar situations, okay? Now, the way that I then break that down in my own classroom, I absolutely guarantee that when you sit down to do your HSC exam, you will be faced with several questions that are taken from a context outside of what you would have covered in the classroom. Now, that's certainly not to induce stress in you, but the idea is that you are able to apply the knowledge and the skills that you've learnt in class to this unfamiliar situation, because it is a key descriptor of a band 6 student. So you should really be expecting questions that you have not covered the content in class specifically, but the necessary content and skills to answer that question will have been covered. So don't get flustered by those particular questions, but just understand that you need to apply that learned knowledge to this now foreign context that you're faced with it. And I'll certainly take you through an example of that as we move through the presentation. I have mentioned this, but certainly 60% of your school-based assessment will have been based on the working scientifically skills and 40% on the knowledge and understanding. Quite often we do these intuitively when we're doing the experiments, but it's really important to understand that, again, assessment questions, and certainly in the trial HSC and the HSC exam itself, these particular working scientifically skills will absolutely be assessed in the trial HSC and the HSC exam. So it's really important that you have a really good and deep understanding of what each of these means and how to apply your knowledge to them. We've covered the tips on what to do in the lead up to your HSC exam. Now, some key tips to put in place during the HSC exam. Teachers, the exam writers, we understand that it is a stress-inducing situation, but remember the following. You have 100 marks allocated within the paper and 180 minutes or 3 hours to do the exam. That equates to roughly 1.8 minutes per mark. Some of those multiple choice you'll punch out quite quickly in 10, 15, 20, 30 seconds. And in actual fact, some of those longer response items, that means that you can allocate roughly 2 minutes per mark. And you can expect a 7, 8, or 9 mark question to appear. And therefore, we're really looking at around 20 minutes that you can allocate to answering that question. And it's really important that you don't spend the 20 minutes allocated to actually just writing your response down. A significant amount of that should be planning a structured response, which I'll come to in a moment. Now, it's worth noting that attempts are made to scaffold the complexity of the paper. This is not just biology, but it's applicable across all of your courses. They attempt to make question 1 easy, and then we move our way through the 20 multiple choice to the more complex questions. Now, that's certainly the intent, and that's just to ease you into the paper. So don't be thinking they're out to trick you. The goal is certainly not to trick you. It's to ensure that you demonstrate the knowledge that you have, but just be mindful that a nice relaxing start to the paper, and as we move our way through, it's going to require a little more thinking and planning on your part to answer the questions. I'll talk briefly in a moment to point number 3. It's essential that you read the questions carefully and cube them, and I'll talk about what cube means shortly. Every word in the question or stimulus has meaning, okay? Look at the mark allocation. Is it a 2-mark, a 3-mark, or even as of last year, we had in 2021, we had a 9-mark question. There will be basically points allocated to each and every one of those marks. So if it's a 9-mark question, you need to be planning, having a think about where those 9 marks are going to be allocated, and again, structure your response accordingly to obtain the most marks that you possibly can. Now, another acronym for you, it's a widely used one, but plan a structured response using ALARM, or another, I guess more the actual acronym that you'll put in place during your exams is IDEA, and I'll talk to that shortly in a moment. And always include examples where appropriate to support your actual answers, okay? Now, another key tip, and I'll take you through some previous HSC questions shortly, never leave your answer blank. That will go down as an NA or a non-attempt. You must write something, and if it's basically anything that springs to mind, if you feel you don't know the answer, put something down, a word, a sentence that actually relates to content associated with the particular question, because in nearly all instances for 1 mark, there is next to that 1 mark some relevant information. So if you don't put anything down, it's an automatic non-attempt, but you're best trying to just accumulate those some relevant information marks in order to obtain marks for those questions that you're not quite certain about. So I mentioned the acronym CUBE. Again, it's quite widespread, but for those that aren't familiar with it, when you're faced with a question, and this is typically applied to all your shorter and longer response questions, try and get into the habit of circling the verb. I mentioned that the significant majority of your paper will be pitched at that explain level, and where students lose easy marks really is where they stop short and answer at the identify or the describe level. So we circle the verb, we then underline the key content of the question as it relates in this case to biology, and then box important information. And again, you'll see this with some of the examples I take you through. Plurals are absolutely essential that you circle, or box in green the S at the end of that, because if they say, for example, with reference to 2 examples, marks will be allocated to more than 1 example. And the word "and" is another really important one, because the question, the marking criteria then will largely be broken into 2 parts: what sits before the "and" and what comes after the "and." And then finally, that second yellow dashed box is where relevant to elaborate with providing evidence and examples as it relates to the question. Okay. Now, the second acronym that I mentioned was that of ALARM. Now, ALARM is the overarching term that responds to a learning and responding matrix, hence the acronym ALARM. But more importantly, the concept is as follows. As humans, no matter what the field is, we learn in a chronological order. Before you can go and give an in-depth explanation or analysis of a concept, you first need to identify what that concept is. We then need to describe it, give some characteristics and features of it before we can explain it, and then before we can move on and do the complex analysis, assessment, or evaluation. So we learn in this order, and largely this idea behind the idea is that we just try and get you to pause and analyze the question before you commit pen to paper, and then think about that scaffolded response. If the question was pitched at the explain level, think about having your opening sentence as identifying the key concept of the question. We then give some characteristics and features of it, and we finish up with the explain. Now, the NESA definition relates to cause and effect. Fair to say, what does that then mean? I like my conjunctions. As a result, this leads to, because of this, and then we follow on as it relates to the question. And then it tells the examiner that you know what it means to explain, and you're giving, hopefully, the necessary depth required. And then what the idea behind this idea is that you stop at the required level of the question. If it was a describe question, we identify, we describe, full stop, move on to the next question. If it was an explain, we move through that scaffolded approach up to the explain, and again, we move on to the next question. When we look at the mark allocation, it will give you a real indication of the amount of information required. Don't go spending 15 minutes writing a really long response to a 2 or 3 mark question. Let's put this all into practice. I'm going to take you through some of the trickier exam questions that I've encountered over the years, or some of the ones that certainly I found quite interesting, and that we can apply some of these concepts I've covered with you in the previous part of the presentation. So this question is from 2014, but it still relates to the current syllabus as has been delivered to you. So scientists have tried to achieve a viable embryo by using 2 ova, that is eggs from the same female. Explain whether the offspring produced using this process would be a clone of the female whose 2 ova were used. And then use your knowledge of gamete formation and sexual reproduction to support your answer. Now, what I've then done is at the bottom of the page, obviously same question, and I've gone and cubed it as I applied my, process to it. Circle the verb, it's an explain. So we need to go into a relatively, reasonable amount of detail in terms of your response. And you'll note what is the key biology that you are required to explain. That word there, clone, it's certainly a very specific word to biology, where we're looking at clone, we're looking at gamete formation and sexual reproduction. And when we look at the marking criteria on the next page, you'll note that all important and that I've gone and put the green box around. I guarantee that if you stop short and only address gamete formation and didn't address sexual reproduction, you're instantly dropping right down the mark bracket too. So we're talking about 3 marks maximum. Even though you've given a really good answer on gamete formation, you must address sexual reproduction as well. Now, the important information that you then need to address as well in the yellow boxes there, use your knowledge. Nowhere in here does it talk about crossing over, independent assortment, random segregation, but it is assumed based on your studies that you know that information. So using your knowledge of those concepts and in order to support your answer to this question. Hopefully you've locked in whether it is or isn't a clone, and I'll now take you through the marking criteria. Maybe not, come on. There we go, okay. So I'm not going to take you through this in a significant amount of depth, except to point out, the key concepts and points to be made from this slideshow. Now, firstly, note you needed to commit to whether this organism was a clone. Now, jumping over to the allocated marking criteria, if you got that wrong straight off the bat, you are way down at a maximum mark of 2, maybe 3, depending upon the remainder of your response. Now, in all cases, the marking criteria will be explicitly linked to the actual question. So at the bottom of the page there, we have a sample answer. And just so that if you follow yourself around the arrows, you'll note that, again, cubed it, the verb is an explain. Now, looking at the marking criteria provided by NASA, for 5 out of 5, it clearly explains the relationship between the structure and behavior of chromosomes. So there's your knowledge and understanding, and the production of unique gametes. So that's the sexual reproduction component of it. So just again, highlighting that if you want to start accessing those higher marks, you need to be cubing the question, looking at the marks allocated to the question, and making sure that your response is scaffolded appropriately. And then the other point to be made at the very bottom of that marking criteria is that for 1 mark, provide some relevant information. Again, that's going to be a theme that's repeated for the remainder of this presentation. So just always remember to commit pen to paper. Writing something, just the words meiosis, wouldn't likely get you 1 mark in this question or for your response. Now, with this particular question, it is from the syllabus that you are studying. I wanted to point out a couple of things in particular. This is question 33. You'll note you'll probably panic when you first see it because it's worth 20 marks overall, but it is then scaffolded. It's actually broken down into a whole series of smaller questions. I think it's reasonable advice to tell you that the bigger mark questions will be a context will be provided that is across multiple modules. In this case, you can see module 5, module 6, and module 7, where the questions actually addressed content from each of those modules. And then down there in underneath the table there, we've got quite an explicit skills-based question. Now, the other thing I wanted to draw your attention to with this particular one, this is a really good example for part A here. Outline the main steps that brain cells use to make proteins such as amyloid beta. Now, it would be reasonable for you to, when you read that question, to think, oh my goodness, the teacher didn't cover that in class. The good news is that unless the teacher had an affinity for this particular area of science, there's probably not a single teacher in the state that would have covered this particular example. okay? But that's not the point of the question. The question is asking you about protein synthesis, okay? It's not specific or necessarily, the focus isn't on amyloid beta and brain cells. It's about you demonstrating your knowledge of protein synthesis. So they're looking for those key terms of transcription, translation. We've got the DNA, we've got the mRNA, we've got the tRNA, and then you fill in the gaps between there. And again, just to highlight that on the marking criteria, here we go, outline the main steps that brain cells use to make proteins such as amyloid beta. So outlining, so that's giving just a series of brief steps, a bit higher than an identify, so you just need to give some of the key steps. And again, note that when you look at the actual marking criteria, there's no mention of beta amyloid or brain cells. It's about transcription, and they even capitalize it for you, and translation. Okay, and then again, amyloid beta, I'm just joining that back to that band 6 descriptor there, where those top students are able to apply learned knowledge to abstract ideas and also to unfamiliar situations. I guarantee many students would have been faced with this particular question. They would have freaked out and left it blank because they would have been angry at their teacher for not having taught them about amyloid beta, not the point of the question. This particular question is from the 2021 paper. I'm just going to take you through part A of it. Overall, it was worth 7 marks, but again, they're always going to be throwing curveballs at you. I love my pedigrees, and I teach my students to always look for specific or certain patterns that will appear that then give you an indicator, whether it's a recessive or a dominant trait or sex link, for example. But this question, I guess, turned that on its head because you had to have a deeper knowledge of of module 6 in terms of mutations and what that would appear like in a pedigree such as this. Okay, so you've been given the pedigree. Is this trait likely to be the result of a somatic or germline mutation? And importantly, justify your response. So 3 marks. So I've attempted to cube the question for you in part. Is this trait, so they're interested specifically in this particular trait, the result of a somatic or germline mutation, and we needed to justify your response. And again, looking at how that question then marries up with the marking criteria, no surprises. Look what we have there. In green, the student for 3 marks needed to justify the type of mutation with reference to that pedigree. And so you need to first up commit to identify whether it was or wasn't a somatic mutation, and it actually is a somatic mutation, and importantly, it is not present in the parents or the offspring of the affected male. And I'm just going to jump back for a moment to this particular question. Just note one thing I just want to draw your attention to. It's an autosomal dominant trait. They tell you that in the stimulus of the question. And if you remember that band 6 students apply their knowledge to unfamiliar situations, I just try and drill this into my own students that if you have two parents that don't have the trait, giving rise to a offspring, in this case the son that has the trait, then the trait being followed is nearly always recessive. And the reason for that is that both parents must be heterozygotes. The dominant allele masks that recessive allele, and then both parents have actually passed on that recessive allele to their offspring, in this case the son. But the question, as I looked at that, if I saw no other information, I would be saying that this trait is recessive. But then in the stimulus, they tell us that it's an autosomal dominant trait. So your mind starts spinning as to what's going on here. And the only way that that can occur is if it is a somatic mutation that has occurred in that particular individual. And the reasons for that, as I've just given you, is that both parent or neither parent have the trait, and it otherwise would have been a recessive trait. So the fact that it's appeared in that individual means that it must be a dominant allele. On to a more recent question from the 2021 paper. I love this question for the following reasons. The dot point in question that relates to this question is a for example. Now that means that in my classroom, in your classroom, classrooms across the state, we can actually deliver this particular dot point in different ways. And as a result, the question is generic enough and all the information needed is contained within the stimulus for you to give the answer to the question. And so basically, it's about sickle cell anemia. It is a genetic disorder. In a family, the parents are both known to be heterozygotes. Now, all of these terms are really important. Again, it's assumed knowledge that you know these terms for the mutation that causes sickle cell anemia. So just going down to the mother and the father, if they're heterozygotes, they have one copy of each of those alleles. And so you'd expect there to be two bands because the DNA profile of the product, there are two DNA, sequences of slightly different molecular weight. So they therefore migrate different distances in the gel. So the couple has two unaffected children and is now expecting their third child. They have had an allele screening test to determine whether the child will have sickle cell anemia. Now, important there, every word's important. If you remember back to the introduction that I gave, two unaffected individuals. Okay, so therefore, child 1 and child 2 do not have the condition, in this case, sickle cell anemia. So what that then means is that this particular trait here must be the dominant allele, and therefore this individual is homozygous dominant, and the child 2 is also heterozygote, because if the trait was dominant, you can appreciate that the heterozygotes would actually express the sickle cell phenotype. And therefore, child 1 and child 2 do not have it because they've both got, in child 1's case, both copies are dominant, whereas child 2 has one dominant, one recessive allele, but the dominant allele actually masks the recessive allele. That therefore means that child 3 is homozygous recessive, so little t, and therefore will be guaranteed to have the condition sickle cell anemia. And again, let's put me to the test and look at the marking criteria. So again, we're using the DNA profile. So again, you must engage with the content or the information contained within that DNA profile. And again, the verb in the question was justify. Okay, guess what the marking criteria is going to contain? It's going to require you to justify your response. Surprise, surprise. And again, if you read through that sample sample answer there, you'll see that the justification I gave is sufficient to actually score at that 3-mark level. And again, justifies using the DNA profile because that's what the question required you to do. And again, the only other thing I'll draw your attention to again here, getting a bit repetitive, aren't I? For some relevant information, you achieve 1 mark. So again, throwing those terms out there like heterozygous, homozygous, if it's not contained in the stimulus of the question, it means that you're likely to achieve 1 mark for your response. 1 out of 3, much better than 0 out of 3, or even better than a non-attempt. I wanted to take you through is again from a very recent paper, the 2021 paper, worth 9 marks. So that's as big as it gets in biology paper, and it reads as follows: transgenic fish can reproduce and pass on the dominant transgene, capital T for trait, and that's really important because you're going to need to refer back to the fact that it's dominant when you actually construct your response. Reproduction for aquaculture is strictly controlled using a variety of techniques in order to protect and preserve biodiversity. Now, an important component of this question is students were absolutely required to know the 3 levels, or at a minimum 2 levels of biodiversity. It's easy. Quite often we talk about biodiversity in terms of that ecosystem biodiversity and the number of different species within an area. Many students, it would seem, weren't familiar with the concept of genetic biodiversity, and that's all about the gene pool and the variation that exists within it, and then the impact that these transgenic organisms could then have should they escape out into the wild. Now, I'm not going to read through these one by one. Suffice to say that some of the techniques are outlined below. There's 5 points there. And at the bottom there, analyze how these techniques protect and preserve biodiversity. Okay, so before I then skip to the next slide, we cube it, I guaranteed analyze. Quite a lot of in-depth information is required, and how these techniques, well, how many techniques are there? There's 5 of them. So guess what you need to do? You need to address each and every one of them and understand what it means to protect biodiversity and also preserve biodiversity. You must explicitly address each of those terms, protect and preserve biodiversity. So I'll leave you in your own time to read through this in detail. But again, at the top there, analyze. So we need to know what it means to analyze. And there I've got that nested definition for you. You need to identify the components within each of those techniques and the relationship between them as it relates to protection and biodiversity. Okay, so looking up at that 9-mark bracket, you can see that students needed to demonstrate extensive knowledge and understanding of the biotechnology related to reproduction and biodiversity. Students were required to analyse the techniques, preferably all 5 of the techniques used, and importantly, they had to address both protection and the preservation of biodiversity. And we'll look at the sample answer shortly, but just wanted to point out some other points. If you didn't give the analysis of each of those 5 techniques, you have instantly dropped down into that 7 and 8 mark bracket. But you can still see at that level, we're still getting the scientific analysis of the main techniques. Now, with those main techniques, I'm just going to jump back to this particular slide for a moment. And when we talk about the main techniques, Technique 1 and 5 are largely grouped together in the sense that both of those techniques are protecting both species diversity and ecosystem biodiversity. So it's all about quarantine and stopping the escape of these transgenic organisms into the, I guess, the natural habitat or natural ecosystems, where they could have quite a significant impact on the, I guess, the original non-transgenic version of this species, but also the greater food web and the impact that the transgenic species could have on those. That then lent itself to techniques 2, 3, and 4, which were largely focused on the preservation of biodiversity. And probably the most difficult one for students to grasp was technique 3, where they needed to appreciate where sperm produced is collected and used to fertilize eggs obtained from wild-type non-transgenic salmon. Very few students were able to understand the point of number 3, and that was actually all about maintaining or increasing biodiversity, genetic biodiversity within the transgenic population. Okay, so bringing in that external elements of the gene pool was all about protecting, I guess, the genetic integrity of the transgenic species. Again, I'm not going to take you through this in detail. I'll leave you to read that in your own time. But again, just note in green there, they've addressed all 5 techniques. Techniques 1 and 5, they're grouped together for preventing transgenic salmon from escaping and protecting that species and ecosystem biodiversity. And then we've got techniques 2 for preventing the transgene gene from being passed on. So again, conserving that genetic gene pool, but then importantly, although not mentioned explicitly, technique 3 there is all about ensuring hybrid vigor of the offspring. Okay, so there's many ways in which an 8 or a 9 out of 9 would look for this particular response. This is just a sample answer. But again, please remember that to break your questions down before you commit pen to paper. So now we've covered some of the key tips for pre and during your HSC exam. How can the A+ series help you? So some of the key, I guess, concepts built into the A+ series is that we provide you with key module summaries with all the important hints necessary to break down the syllabus dot points and outcomes. There are concept maps for each of the core modules, and also for the skills section of the course. And leading on from that, we've got the Working Scientifically chapter, which really does focus on those important skills which you absolutely will be assessed on in your trial and HSC examination. We then provide you with revision summaries, which are provided to really consolidate your understanding where you've read through the chapters, you've got all your other relevant information together, and you can put pen to paper in the back of the book and make sure you really understand the core biology that you need to know for each of your modules. And then we've got the exam practice questions. And this includes a range of NESA HSC questions and also HSC style questions with solutions and explanations that have been written by the authors. Finally, we have the online resources, which includes the glossary of terms and flashcards where you can test yourself to make sure that you really do understand those words that are uniquely biology based. New in 2022, again, I'm the biology author, but many students don't stop with biology. They'll certainly engage with physics, chemistry, and biologies. And the good news is that we have resources for each of those sciences. Where to next? So for those that are interested in the resource and want to excel in their HSC exam, I'd strongly encourage you to visit the following website for all the relevant and necessary information needed to find out more about the product and purchase if you choose.